Good afternoon, Dr. Okapodu. My name is Danielle Gray, and today I'm presenting my project on New World arenaviruses and their diversity from Old World arenaviruses, specifically doing my topic on Sabia virus. Now, I chose this topic because currently, for 2 years, I have actually been a lab assistant in Woods dealing with microbiology. And I deal with a lot of bacteria, E. coli, pyrogens, everything like that. And I actually culture and I also clean hazardous waste. So I did this topic just because, number one, I really do like learning about microbiology, and I like learning about viruses. I have just gotten into more just looking into pathogens. And also, the fact that I want to be a mortician, that plays a part in the fact that I kind of like gross bacteria. So that plays a huge part in why I actually did my presentation on this pathogen, Sabia virus. Sabia virus is an arenavirus, and an arenavirus is basically a family of viruses whose members are generally associated with rodent-transmitted diseases in humans. It is zoonotic, meaning that the only reservoir or the only thing that gets this virus is actually rodents or mice, mosquitoes, anything like that. As you can see on my picture, I basically have a picture up here that basically shows the viruses, arenavirus specifically, in certain regions of North America and South America. As you can see, majority of South America actually has a majority of the arenaviruses. You can actually see Sabia virus actually right here in the corner. There are also two arenaviruses, one on the East Coast and one on the West Coast, that are also predominantly in Florida and in California. There are two groups of arenaviruses. There is Old World and there is New World. Old World viruses are located in the Eastern Hemisphere. They include Europe, Asia, Africa, and the Americas. The reservoir is rodents, mice, and rats. New World arenaviruses, which Sabia virus is under that, is located in the Western Hemisphere. It includes Trinidad and areas in South America. Also, its reservoir is rodents, bats, and mosquitoes. The virus that I basically focused on was Sabia virus. There's numerous amounts of arenaviruses actually out, stemming from New World and Old World. Sabia virus, known as Brazilian hemorrhagic fever, it was found in the 1990s in Sao Paulo, Brazil. It is a New World virus. As I said before, is a biosafety level 4 pathogen. It's highly lethal and infectious. Human infection comes from an individual in contact with excretions of an infected rodent, contaminated food, or inhalation of tiny particles soiled with rodent urine or saliva. The first infection occurred in a woman located in the village of Sabia, Brazil, and its incubation is between 7 and 16 days. This virus is an aerosol, which is plays a big part in the fact that it can be spread through haze, clouds, rain, dust, anything like that. So thinking of the area that is mostly focused in, South America, there's a lot of dirt roads, a lot of villages. So that plays a part in the fact that this virus can be spread. Some of the symptoms from this specific hemorrhagic fever are, of course, fever, headache, eye redness, fatigue, diarrhea, also blooding underneath the skin and internal organs, nervous system malfunction, seizures, and it can even lead to a coma and even death. But from the virus, that is not the main cause. Usually it's from the symptoms. Old World versus New World arenaviruses. The evolution of these hemorrhagic fevers are changing the outlook on health and disease control of the U.S. due to cross-contamination, genetic variation of T cells, and biosafety/bioterrorism. Arenaviruses have been changing geographic locations, and with that factor, they are becoming more and more diverse. The CDC, Centers of Disease Control, actually has a whole section about arenaviruses. They have actually been trying to focus on it because these new world viruses are stemming from old world viruses, which plays a factor in the fact that they are changing and it's becoming more hard to keep public safety health-wise, to make it easier for people to get vaccine or harder to get vaccines, as you will see later on. Finding an article in the Journal of Virology, Comparative Analysis of Disease Pathogenesis and Molecular Mechanisms of New World and Old World Arena Infections, 2013, Old World and New World natural reservoir hosts can maintain high viral loads, thus not being affected by the virus as much as humans. Old World and New World arenaviruses trigger different immune responses affecting T cell response. When you get a cold or when you have some kind of disease, the automatic, you have 911 cells, of course, and you have your 911 cells basically are the first ones to attack. They are macrophages, but they actually die very quick. Your T cell response is the reason why you have symptoms, whether it be inflammation, whether it be a cough, a fever. Those are all T cell responses. You have T helper cells, and these cells basically can help with trying to fight the disease, fight the pathogen, whatever it may be. It can either be a positive or it can actually be a negative. Although there are some treatments and vaccines for some of the virus present, most are too complex to come up with a vaccine due to their destructive nature. Those infected may share the same arenavirus species but show varied disease manifestations. Saying basically not one case of the Sabia virus has been the same for everybody. So there are 3 known cases. One person has died from this virus. Depending on location and incubation time of the virus, death is inevitable for some. The first case that I actually looked at was actually in Connecticut, is arenavirus infection. Basically, by the Source Center for Disease Control, a senior level research scientist was actually purifying this virus, but in a centrifuge tube, but the centrifuge tube had broke, and it was transported through aerosols, which basically in the air, and this person contracted the virus. Now, this virus that they were actually working on was the first legit research virus from the actual first person who caught this virus. And testing had proved that his white blood cell and his platelet count had declined extremely. Further investigation, in the Connecticut Department of Public Health and Addiction Services, the virologist was working alone and exposed by aerosol, basically contracted the virus, and those who came in contact were actually surveillance and showed no signs of secondary infection. So they actually quarantined this person and actually put them in a low pressure room where he was separated from the rest of his colleagues in order to see would he have more symptoms of the disease. There was also a Sabia virus incident at Yale University in 1994. One of the committees containing experts in virology outside of Yale cited a number of factors that could have led to the accident, including a lack of oversight and consultation by university management. Basically, these places that were doing this research for the Sabia virus, it had found by the external committee headed by Dr. Clarence J. Peters of the Federal Centers of Disease Control and Prevention in Atlanta, stated that if the Yale team had conducted appropriate risk assessment, then this would have never happened. And it also determined that there was a lack of appropriate biosafety training. When it comes to T cells, in the frontiersign.org, common antiviral cytotoxic T lymphocytes epitope for diverse arenaviruses. Basically, in this article, I found that there was a comparison between Old World and New World viruses, that they share several amino acids, but as I have said before, the symptoms can be different. And they also found that some Old World and New World arenaviruses have a same common function, nucleoprotein epitope, which allow for vaccines to be made and offer immune protection. What scientists and researchers are trying to do, or trying to find a vaccine to cure these arenaviruses. They have actually found 2 vaccines now, but Sabia virus is actually not included in that. More about Sabia virus and its biohazard safety level. It is a level 4 pathogen, and as you can see, all the way at the top at the pyramid, that's where it stands. It's the smallest, but it's the most deadly of viruses and disease that are located there. BSL-4 builds upon the containment requirements of BSL-3, which is basically laboratory requirement practices. Laboratorians are under medical surveillance and might receive immunizations for microbes they work with. Access to the laboratory is restricted and controlled at all times. It is the highest level of biological safety, and the microbes are very dangerous, exotic, posing a high risk of aerosol transmitted infection, so transmitted through the air. This leads a bad rep to people saying with bioterrorism, just as bad anthrax is, there are people who are studying this virus and are actually using it as a bioterrorism threat to, whether it be the United States or other countries. Because it doesn't have a vaccine, there is no cure per se, this can actually be a very high risk event if it were to happen. Just like I said, as much as anthrax was very important, this is very important also. Many people do not know this, so lack of knowledge plays a big part in the way safety is used against bioterrorism. Centers of Disease Control and Prevention wrote an article about managing potential laboratory exposure, specifically to the Ebola virus, but Sabia virus is also included in that because Ebola is a level 4 pathogen. Basically, the USAMRIID, which is the United States Army Medical Research of Infection, and the Medical Containment Suite, which is the MCS, basically confirmed the quarantine of those who were exposed to biohazard level 4 pathogens. Basically, it was stated that these 2 specific groups, basically came out with a law saying anybody that is exposed to any level 4 pathogen actually has the ability to be quarantined without question. And actually, the MCS created a 9-step process in order to contain the possibility of an accident occurring again. As you can see through steps 1 through 9, such as prepare, assess the patient, gather appropriate consultants, conduct appropriate logistics, determine the treatment, keep a journal, anything like that in order to prevent this from happening again. More about biosafety, level 4, laboratory practices. These people, as you can see in this picture right here, you can see that these people are well suited. They have their own air chambers. The air and the water that does come into the facility in this specific room actually goes through a decontamination process. All the work with microbe must be performed by wearing a full body suit, air supply, positive pressure, a suit. The facility construction, majority of these biosafety or biohazard labs are in the United States. There are some in other countries, but the United States has the most. There's actually one located in Maryland and Atlanta, just to name 2 that are very close to the Virginia area. These Labs are dedicated with supply and exhaust air, as well as vacuum lines and decontamination systems. They are actually kind of like rooms within a room that's within a room within a building. So, as far as more about T-cell response in Journal of Virology basically said it plays a critical role in providing protection, immunity following infection with the oral arenaviruses. Basically, these epitope-based vaccinations are used in order to protect against multiple arenaviruses. Even though some of the arenaviruses were not 100% sequenced, it was found that there was a link between some arenaviruses producing a vaccine. Sabia virus was not a candidate, but just recently, an article published in the Indian Journal of Pharmaceutical and Biological Research just in the month of February was approved that the Sabia virus glycoprotein G1 is a perpetual membrane protein involved in viral absorption and pathogenic infection. And basically, from that protein, vaccines were being made or trying to come about to be made in order to cure in order to cure the virus. There are others candidates for possible vaccines also, but this article basically stated that it is in the process, these researchers are in the process of coming up with a legit virus in order to give to people to these countries that do not, i.e. believe in vaccination or cannot have vaccinations at all due to money-wise or don't have the proper health care. As you can see in my table, the virologist that I had talked about before who contracted Sabia virus, basically, he was put into a low-pressure room and had multiple tests were done on him. As you can see, on day 1, you can see right here. You can see that his blood, his white blood cell count and his platelet count had decreased tremendously. You can also see how his temperature was 103 degrees at one point in time, all the way down to day 10 where he started to feel better and his temperature came, lower down to his regular temperature, 98.6. Also, his white blood count had also came back in order, and his platelet count. You could also see that his calcium had went down, and his calcium had went up actually, then had went down, and other specific minerals and enzymes that were in his body that basically deteriorated from day 1 and then got better at day 10. Overall, studies show that CDC has been furthering the study of prevention and safety of human health when it comes to highly pathogenic diseases. Arena status warrants further investigation into their pathogenesis and immune response in their biology. McLeay, Lang, and Lai, 2013. Available literature and articles do not confirm an improved cure for Sabia virus, but has for other hemorrhagic fevers. Even though arenaviruses do share the same characteristics, it is known that these viruses are changing. Depending on their location, the fact of cross-contamination, the response to T cells, the response in T cells, and also the safety, the world has not came up faster enough to protect the general public. So in order to do that, the CDC is trying to prevent, trying to come up with new research, trying to have harsher laws when it comes to biosafety. And this is actually, starting to get better as technology is improving. So is the ability for public health to go up more. And hopefully one day there is availability where these arenaviruses do not come in contact with people and do not harm people, and we're able to just give them an easy vaccine and go about their day. And that is my project. I hope you enjoy, Dr. Okapodu. Thank you.